Good morning, everyone. And as I did yesterday, as I invite today's speaker to our event, I also would like to extend a warm welcome to all our viewers from all around the world and express our deep gratitude for the hundreds of registrations and paper submissions that our 9th Biology Week has received so far. And as I also said before, these huge numbers are certainly a reflection of the level of effort and commitment of our team and collaborators to making the event happen. So it is in the spirit that I extend a special welcome to our guest today here, Dr. Cromwell Purchase, whom I gladly invite to join us to present his lecture entitled Extinction of the Spix's Macaw and its Meandering Journey Back to the Caatinga, Brazil, a story filled with innovative approaches, merging multiple scientific disciplines and community integration to reach the end goal. Dr. Purchase, welcome and thank you very much for being with us today.
Thank you very much.
Yeah, I'll just read the
presentation. Okay.
Yes, it's there. So before he begins, Dr. Cromwell Purchase is a South African. Cromwell has bred and managed avian and zoological collections, including his own from a young age, and has always had a deep interest in wildlife, the environment, natural habitats, and particularly birds. After school, Cromwell studied human medicine, physiology, biochemistry, and molecular medicine and medical biochemistry degrees. He went on to do a master's in veterinary science, focusing on avian medicine, diseases, and vaccine technology, following that up with a PhD in avian nutritional physiology. Having completed his studies, Cromwell followed a long-time passion of wanting to work with Spix's macaws and moved to Qatar to manage the Blue Macaw project for the royal family of Qatar's wildlife preservation Al-Wamra, and became the international stud bookkeeper and genealogist and project management consultant for the Spix's Macaw recovery project. After a few years, he became the director of the facility, and after the passing of the founder, he managed the facility until its closing in 2018. When he moved, along with all the Spix's macaws and other species, to the Association for the Conservation of Threatened Parrots in Germany. As director of the German facility, he continued to manage the largest population of Spix's macaws on the planet. Later, Cromwell and his wife moved to Brazil to prepare the inhabited breeding and release facility for the arrival of birds and spearhead the in-situ release project. So, Dr. Purchase, again, thank you very much. And you have up to 90 minutes for your presentation, and the last 30 minutes will be reserved for interaction with our viewers and for answering any questions. So the floor is yours. Thank you.
Okay, thank you very much. Hello, everyone. Thank you for coming to listen, and I hope that you'll get something out of this presentation. The Spix macaw has meant a lot to me, and I've dedicated a large part of my working life to the Spix macaw, and hopefully it'll be a great success. The extinction of the species is something that was a devastation for many people that followed as the species was declining in the wild. And I'd like to show you a little bit of the history and how the project has moved forward and where we are today. So the Spix macaw comes from the northeast of Brazil, in the state of Bahia, and the local town is called Curaçao. And this area is unlike what the Hollywood movie Rio makes it look out to be. It's not, this bird does not come from the Amazon jungle. It's not a forest species. In fact, it comes from the Caatinga, which is an arid environment, a semi-desert, if you will. And these birds inhabited the creek lines. So you would get large trees in the creek lines. And then if you go 50 meters beyond these trees on either side, it's small shrubs, and there's very little available. And unfortunately, these arid zones are very easily degraded by farming, especially livestock farming. So it's been very difficult for the species to continue its integration and grow in the wild along with other species. And unfortunately, the Spix macaw seems to be a species that has been on decline for quite a while. A little bit of a history, when von Spix was doing his expeditions in Brazil in 1819, he came across a very small group of Spix macaws. And even back then in 1819, he considered the species to be rare in the wild. especially compared to all the other species that he had seen as he traveled and went on his expeditions around the country. So even in 1819, the Spix macaw was an endangered species in what we know of. And after von Spix had his discovery, and the Spix macaw was then known to science, the Spix macaw was not seen again by biologists or by educated people till 1986. And even the local communities, most of them didn't know where the Spix was, other than obviously a few people who were exploiting the species. And that is the wildlife trafficking, which was rife in the 1960s and 70s in the region. And the wildlife trafficking is still around today, although a little bit less than what it was back then. But for the Spix macaw, it was the 60s and 70s was the time when trafficking really took them out of the running for a wild population and brought them to their knees. But habitat degradation in the region is something that has been going on for many, many years before the Spix became extinct. And that decline shown in 1987 when the last 3 Spix macaws disappeared, and it was shown that wildlife trafficking had disrupted those last 3. There was one known breeding nest, and that breeding nest was taken out by some poachers in the evening. They cut open the side of the tree and took the took the female out and broke the eggs in the process. And that was the end of the Spix as we knew it in 1987. So only 1 year after their discovery, those 3 that were found disappeared. And it wasn't until 1990 when there were rumors that there was the sound, the voice of the Spix in the creeks of the Caatinga. And that information brought biologists out to go and have a look, and they found only one, a single male Spix macaw, and he was the last of his species. And quite aptly, the Spix macaw is unlike any other any other macaw, and it has a genus of its own, Cyanopsitta, spixii. And this last male, ironically, was the last of his species, sitting out there for a decade. And halfway through that decade, in 1995, there was quite a controversial issue, and that was releasing a female to this male. Many believed that this was a mistake, and that in fact the last male should be caught and brought into the captive program. Because as all of this was going on in the wild, there was a group of birds in captivity, and obviously from smuggled offspring that had been taken out of the wild in the 60s and 70s and made their way into the captive population. But what the Brazil government managed to do is they managed to get these people involved in working towards a captive population that would allow for a future release of Spix macaws into the wild. And in doing that, a project was created to do exactly that. And this project, the people involved in this project were the ones that were now considering releasing this female along with the Brazil government for this male. And it was an interesting choice because the male that was alone in the wild for so long, he had mated up with a maracana, which another name for it is the blue-winged macaw, another small macaw found in the region that also loosely flocks like the Spix macaw does. And the idea was that if you release this Spix female, that he might not even consider her because he had already been bonded to a maracana. However, the release went forward, and in May 1995, the female was seen flying with the male, as well as with the maracana. So the 3 of them were flying around together. But sadly, by June, that female disappeared again. And again, that male was alone. And he would be alone for the rest of his time until October 2000, when the news hit the world. Everyone who had an interest in Spix macaws and were following the project got hit with the news that the last Spix macaw was gone. The Caatinga no longer had the voice of the Spix macaw in it, and it was lost once again. So this whole story and process of how the biologists and everyone involved were pushing to try and help get this bird back out into the wild, just basically came to an end. It came to a crushing end. And it made the Spix macaw captive program so much more important, because all of a sudden it was the last hope for the Spix. And with that, I show you some numbers. And when that last Spix macaw disappeared in the wild, there were 53 birds in the captive population. And when I started, well, then 5 years later, there were only 54. So there was a net increase of 1 bird in the population over those 5 years, which sort of gives you an idea of how difficult it was to breed these birds in captivity. And it wasn't because they weren't willing to breed. It was because there were fertility issues and there were disease issues. So there were also mortalities at the same time. So you would get hatches and you would get mortalities. And obviously this process continued. But the first 5 years, the net increase of 1. The next 5 years, it increased a little bit more. And again, between 2010, I started in a project at Al Wabra in Qatar at the end of 2010. And when I started, you'll see the little pictures on the side. I had a population, I inherited a population that had a diamond as a pyramid. So the population pyramid wasn't the pyramid it should be. It was a diamond. And we basically, our goal, our main aim was to move this from a diamond back into a pyramid again. And what we did was we worked really, really hard on a whole lot of different research concepts in order to come to a point where we actually saw positive change. And you can see the first 4 lines are all 5-year intervals. And then it becomes a 3-year interval and the increase of 50 birds, a 2-year interval, and now a 1-year interval. And we're very happy to say that 2021 has been a remarkable season, where we already have 48 chicks for the season, and the season's not over yet. So the exponential increase that you're seeing here is a direct relation to a lot of hard, innovative research that was put together. Now, one of the most important things that we found is obviously there were problems involved, problems that we had to overcome. And eye color was something that was very, very interesting. So there were 2 populations in the world when the Spix went extinct in the wild, 2 main populations. And the one was in Switzerland and the other one in the Philippines. And the birds in Switzerland had a white eye, and the birds in the Philippines had a black eye. And these 2 populations were bought out by the Sheikh in Qatar, and they were merged at the facility in Qatar. And one of the big problems associated with merging the population was that you had 90% of the world's population of Spix in one place. And this was a concern for everyone. Originally, it wasn't the plan. The plan was to keep a population in Switzerland under the management of the Sheikh and the Sheikh staff. But there was an attempted break-in at the facility in Switzerland. And it was decided that those birds would be moved to Qatar. Now, it was quite a controversial move. Everyone was worried. 90% of the population of an extinct in the wild species in one place. There are pros and cons. And unfortunately, the cons were massive at the time, because the birds that came from the Philippines with the black eye, that black eye was anthracosis, which was caused by air pollution and heavy metal poisoning. So we had inherited these birds from the Philippines that, in theory, would have major problems with regard to fertility. Heavy metal poisoning, especially lead, is a serious issue for male fertility. So with a population that is already inbred and already showing signs of infertility issues, adding this concern created even more issues, along with the fact that we had a disease outbreak in the Swiss population that was realized a few years later to be bornovirus. Proventricular dilation disease is the disease that comes out from the bornovirus. And fortunately, the pros to having the birds in one place was being able to manage and research them and allow that research to move a lot faster. So when you have all the birds in one place, your research can progress at a much faster rate because you have the largest number possible to do the research on. And this actually became a crucial thing. Fortunately, the management, at the time when these birds arrived in 2004, these two populations between 2002 and 2004 at Al-Wabra, it was managed by a man by the name of Sven Hammer, who was a German. And he had brought in a plan in order to quarantine off sections of the population on the same facility, having different staff, and what he managed to do was he managed to manage and control the coronavirus outbreak. And he was able to then incorporate this protective biosecurity in order to save the population and allow for that population to continue. Now, this diagram is a horrible diagram, and don't worry about the numbers. All I wanted to do was show you, with the microsatellite work that was done at Sao Paulo University for the genetics in order to help us with the pairing options. It goes from blue to red, whereas red is very bad genetic compatibility and the blue is a much better genetic compatibility. And all I wanted you to see in this picture was that main aim was to try and get as many blue pairings as possible. The males on the x-axis and the females on the y. And it was just to try and get as many genetic combinations that were good as possible in order to keep a genetically viable population. Now, the research that was needed included hormone, egg, and sperm analysis. It included artificial insemination. It included in vitro fertilization and genome sequencing. I'll run you through them quite quickly, as it can be quite boring. So, with the reproductive research, we have the different hormones that you need to test. And we were testing stress hormones and reproductive hormones. And one of the key factors that this brought to our attention was we were testing in season and out of season between successful breeders and unsuccessful breeders, between birds that were not breeding and birds that were breeding. Now, with regard to the successful and unsuccessful, it's fertile eggs and infertile eggs is what we're more talking about. And if you look at the diagram that we have, the green triangle is fertile eggs, and the purple triangle is infertile eggs. The blue and yellow lines are the related reproductive hormones of the male and female. So when the male and female were hormonally in sync, they produced fertile eggs. When they were not, they did not. And the reason is quite simple. So Spix macaws, the males only produce semen for short periods of time, because the one thing about a bird is it needs to be efficient in its energy because it has to fly. So in doing that, it doesn't waste energy in producing things like semen where it's not needed. So Spix only produce semen in the breeding season when they are stimulated by the female. The problem with that is that if your female is not synchronized with you hormone-wise and behavioral-wise, you potentially will have a mating occurring when the female's hormones are not at the right level to produce fertile eggs, or the male is not actually producing any semen. And this was a crucial aspect of the research and a little piece of the puzzle. Now, all these little things that we were doing and all these little tests that we were doing, they were all pieces of a puzzle that combined together created the outcome that showed this exponential growth in population. Now, the egg analysis was another very important piece in the puzzle. Fertility was a major issue. With a 10% fertility rate, we were finding that most of the eggs were visibly infertile. The problem is that when we did the research behind it, we found that actually most of those eggs were fertile, but it was extremely early embryonic death. And that early embryonic death was so early that the egg didn't that it had been fertilized. So the embryo had died before that process had got to the point where you could see any fertility in the eggs. So it didn't mean the eggs were infertile. And with the fluorescent microscopy, where we took off the perivitelin layers, I went to Sheffield University and learned from a very good professor there how to do this technique and incorporated it into the project. And there, we removed the 2 perivitelin layers of the egg, and you could see under the microscope the holes in the egg, and it would dye so that it would show the sperm and you could see that actually sometimes there was sperm between the two layers, but they didn't actually get into the egg. So the sperm was getting all the way to the egg. It was getting to the perivitral layer, but then when the second layer was put on top, it hadn't gone through, and it hadn't made it to the germinal disc where fertility needs to actually happen. And then you were having eggs where the sperm was getting through, but then it was getting to the germinal disc, and you were getting slight proliferation of nucleated cells, but then it would stop. And the idea was that potentially the semen's energy had run out by the time it got to that point, and multiple other factors, but these were things that we had to look at. But it also gave us the idea that the actual genetics, the genetic combinations of some of these pairs were not good pairings, and it might have been fatal flaws in the genetics that was not allowing this development to continue. So another piece of the puzzle, along with the hormones, brought us to the next point. We were looking at sperm and feces. This would allow us, so just collecting fecal samples, so non-invasive, no disruption to the birds themselves, in order to find out the point when the males were actually producing semen and inseminating to the female. There we go. And this kind of information all brought us to the point where we could start collecting semen. We started with trying electroejaculation, and we were getting the semen out. We were then looking at the morphology, the viability, quantities, qualities, and we could then see all the problems that were associated with the semen that some of these birds had. Some of them had slight variations in the semen that were not viable for swimming. So some of them had two tails, which didn't allow them to swim in a good way, slowing down the semen and not allowing it to get to where it needs to be in time for fertilization to occur. So this became quite an important process to the next point, which is getting these birds ready for artificial insemination. And as we were moving forward, getting the electroejaculation perfected, monitoring the birds on cameras to see when the eggs were laid, etc., when the right time is to inseminate, all these things, we were then moved on to starting the process of artificial insemination. And we were doing direct insemination. So basically what it means is we would be monitoring the cameras and that were in all the nest boxes of the birds. And as soon as an egg was laid, we would then have already a list of which males genetically were the best for that female. And then we would go to those aviaries, we'd catch the male, we would try and get semen from him. So we'd go to the number one male. If he didn't have semen or had too little semen, we'd go to the number two, number three, etc., until we had enough semen to do an insemination. And then we would go, and sometimes we wouldn't have enough from those three males, and we'd have to pool the semen of the three males and inseminate with a pooled semen sample. This work allowed us to figure out what it took in order to get a successful insemination that actually created a chick on the perch. And so what we were doing was we were grading these inseminations, and it basically allowed us to get to the point where we knew exactly how much semen we needed. Any less than that, and the insemination was likely not going to be viable. And anything above that, and we were pretty safe to have about a 90% chance of a successful insemination. And so basically our steps moved us in the direction where we were able to use females and get females to produce that had never produced before. We were able to use males and genetics from males that had also never produced before and move them into this population so that our genetic population grew. And these inseminations allowed us to push forward the next step, which was creating this population pyramid of viable offspring that will help produce the next generations. Because part of the process of semen collection was finding out that a lot of the birds had very little semen, and the Spix macaw has the quantity of semen is much less than other parrots of its size, and so it's something that we had to really look at. And one of the things that we also then pushed forward to, it was the next logical step, was cryopreservation, because we were doing all this work at Al Wabra in Qatar, and the thing is, there were also other facilities in the world that were holding Spix macaws. So the idea of being able to preserve semen that we had collected and send it to a facility that was close to, the pair was close to breeding, and allow for them to do an insemination there, was the idea. We started the work, we got the process, we managed to get some fairly good samples, but this trial was stopped before we actually got to the end result, because we were at the point where we no longer needed the artificial inseminations. We had created the population, the base population of genetically different bird individuals that we needed in order to allow the population's growth to continue and fertility of the population to increase. Now, in vitro fertilization, this is just another idea that we had, because when you are waiting for a bird to lay an egg, it lays its first egg of its clutch. And we didn't inseminate before that first egg because you were never sure when that egg was going to be laid. However, when that first egg is laid, it creates a stamp to then extrapolate your insemination to have a fertile second egg and then a fertile third egg. So that first egg invariably was infertile. So our idea was to try and inseminate an already laid egg. And so we played around with some in vitro fertilization because the egg was already lost. There was no hope for it already. So we weren't losing anything in trying. And we played around with it. We had a few failures. We had a few, well, they were all failures because we never got a chick out of it. But it developed to a point where we were actually getting some good results. Now, this was something that we never thought was possible. Once an egg is laid, it's thought that everything's sealed off. The plan, it's done. The germinal disc is now in process, and the hormones and everything and enzymes have all been in play. But it does seem from the research that we did that it is possible. However, antibiotics need to be put in because there's always a chance. An egg is a host for disease. So there's always a chance that you're going to get an infection. And so there's a lot that still needs to be done on this aspect. But now that the artificial insemination has stopped, we no longer need to move forward with this. But this is a possibility for other projects in the future. Now, with the egg analysis that we were doing, we realized that the genetic data that we had, which we got from Sao Paulo University, which was great for the project. We were able to do, with the microsatellite work, we were able to at least get an idea of which pairings were best. But the egg analysis did show us that we did have pairings that potentially were not good pairings, that seemed to be reasonable pairings on the genetic analysis from the microsatellite work. And microsatellite work, if you know much about genetics, it's a small portion, a small gene that you work across in order to see variation. And it's a gene that shows variability, but doesn't necessarily mean that they are good options reproductively. There is always a possibility that there is another sequence somewhere that is creating the issues. So what we did was we collaborated with Weill Cornell University in Qatar, and we did the genome sequence of the Spix macaw and most of the Spix macaw population. And this allowed us, it gave us the information that where we could look at different parts of the genome sequence, and we could find a better overall match between pairings. This gave us information that for the stud book that allowed the young birds that we now have, that we created this pyramid for, the opportunity to have less infertility and more fertility rate, etc. And it also opened the door for future research. For example, gene expression could possibly be brought into this population or other populations that have a low semen count. By overexpressing semen production genes, there is a possibility that we could have increased semen quality and quantity in the Spix macaw or any other species. So it opened the door for future research. Now, with breeding birds in captivity, there are often issues, and egg deformities, often a problem, malposition in eggs, and sometimes egg yolk lack of absorption. But also one of the key issues is behavioral issues, Spix macaws. Some of the birds have mental behavioral issues, which can be a problem when you have pairings, because those behavioral issues could impact the actual pairing of the birds and thus the egg production or breeding production of that pair. So deciding which birds you're going to put with which birds, how you're going to manage those behavioral issues was quite an important thing. And we've done a lot of behavioral research in order to limit as much the behavioral issues as possible, because one of the things we were worried about was what if some of these behavioral issues are genetic, and breeding this bird then brings that behavioral issue into its offspring and thus perpetuates the problem in the future. So there were a lot of aspects related to behavior, and we've published a few articles recently regarding the behavior of Spix macaws, and it has given us a lot of insight into the population and how to manage them for breeding. Breeding management and egg management is extremely important, and the facilities set up are crucial to ensure that these birds are breeding properly and monitoring the birds. So you basically got a population here, a captive population that has been in Big Brother for the last decade and a half, where every part of their lives has been monitored. They've been watched doing everything in order to try and find all those little things that combined will help the population grow. We had cameras everywhere, monitoring nest boxes, monitoring the aviaries inside, monitoring the aviaries outside, sitting day and night, monitoring to make sure that when the breeding season is on, we get every insemination possible during that insemination period. And the results show. In science, it's rare that you actually do research where the results actually manifest in a productivity increase in the real world. And we were very fortunate to be in a position where we had this, where as we were doing the research and as these pieces of the puzzles were being put together, we were starting to see the changes in real time. We were watching these birds' numbers increase and the hatchability increasing, the numbers of pairs breeding increasing. The fertility rate is becoming much, much better now. And it's awe-inspiring for the researchers involved. It's something that motivates them to push forward and keep going. That positivity is incredible, and that picture just basically says a lot. That's a picture that was taken 2 months ago of this breeding season. And it just sort of gives everyone that bit of support, knowing that what they're doing is actually making a difference. To give you a bit of an idea, so the overview of the population at the moment, most of the population is still at ACDP in Germany, but now that we are processing and moving things around, we're trying to separate populations so that we don't have everything in one place. And so slowly but surely, we're moving forward with infrastructure changes, facility increases in order to get to that point, which I'll get to a little later. But now to just give you, now that you've got a bit of the history, a bit of the science that was done, to give you a bit of background of the facilities themselves. So Al Wabra, Qatar held the biggest population of Spix macaws, 90% of the population for many years. In 2014, the founder of Al Wabra died and his estate was frozen. And this brought us to the point where things needed to change. And the children of the late Sheikh, they understood this, and they understood that there were going to be major problems clearing the estate. And in fact, the estate took more than 5 years to clear because it was so massive. So what we then had to do was we had to figure out how we were going to move 120 Spix macaws from Qatar to Germany. And fortunately, there was a good collaboration for the years before between the facility in Germany and the facility in Qatar, where birds were swapped for breeding purposes, etc., on a regular basis. And so that helped with this process. And in this process, we did, as the stud bookkeeper, we had to get the birds out before the summer started. So we were really pressed for time because the Qatar summers are harsh, and to have birds in boxes, etc., makes it very difficult. And as a stud bookkeeper, I had to manage those shipments because the birds left in 5 separate shipments. And I had to make sure that every shipment had every portion of the genetic viability that we had available for the population. Because if something happened to one of those shipments, we couldn't have an entire genetic pool gone. So it was quite a hair-raising time. It was a difficult time getting everything in place, but the shipments were successful and without incident. Breeding these birds has been very important, and getting the birds to the new facility was also crucial. But one of the crucial aspects of this population is that the Spix macaw was a very, very special bird. It was a very rare bird, and as such, every individual was hand-reared. Every egg was pulled, every individual was hand-reared for decades. And this created a population of hand-reared birds. Now, we were all about conservation. We were all about the plan. There was always an end goal, and that end goal is to see the Spix in the wild. And unfortunately, you've got to be careful of how you hand-rear a bird that you want related to the release of birds into the wild. Now, there are 2 ways of hand-rearing birds. You can hand-rear them for conservation, or you can hand-rear them as a pet. Most people who will hand-rear a bird of the size of a Spix macaw take about 150 days to wean them, whereas we needed to wean them like the parents would. So monitoring the birds and making sure, seeing what the parents are doing, and other species very closely related in regard to size and being a macaw, we would see how many days it took for them to wean, how they were able to get the chicks to the weight that they needed to get to in the time that they were doing it. Because one of the most difficult things to do is to mimic the weight increases of a bird with what the parents do if you hand-rear it. And it's something that we managed to do. And we managed to get the birds increasing in weight very quickly, getting them to the point where they were able to wean between 70 and 90 days, which created a wilder bird. It created a bird that once we put them into the large flocking cages with other Spix macaws, they would integrate quite well. They were more keen to learn because they were out a lot quicker. They weren't sitting in a box for 150 days. They were out. They were able to learn in that period when their mental acuity is so strong. But the end goal is parent-reared. And we're very fortunate that at our Wabra and in Germany, the first parent-reared Spix entered the population. And these chicks were obviously then incorporated with the hand-reared ones in order to help teach them, etc., with some adult birds in the flocking cages, in order to create an environment where you have birds that are not dependent on humans all the time. Now, with this change of facilities, we needed 120 birds moving from Qatar to Germany. Germany needed new cages, aviaries. So they had to build an entire wing of aviaries to house 120 Spix macaws, not to mention a few other species that did come. And flocking cages that allowed for large numbers of birds young birds to be incorporated together in a flock to learn from each other, to have more of a community education. So what we would do is we would have a few birds from previous years in the enclosure as teacher birds, and we would move out a lot of the others and move in all the new youngsters to learn from those birds. And this would create an environment that we're in now in Brazil, where I'll get to that a little bit later, but where we're now getting them ready for release. But these facilities, as you create the facilities, you need obviously a veterinary center, you need a nursery, you need all the different aspects that go with such an important species. You need to be monitoring every possible sign of disease, etc., especially when you've got so many birds in one facility. Monitoring them on cameras, the food, the cleaning of the bowls, everything needs to be managed so that you don't have disease spread. And with this whole idea, and always been the plan to spread the population and not have too many in one place, we managed to have a new partner join us, Paradisa Foundation, which is in Belgium. And they built facilities in order to ensure that we could move a number of the population to them and have another viable population to just save the potential population if there was a disease outbreak. Now, getting to the next stage where the aim was to get these birds to Brazil and the release facilities. So basically what we were doing was creating a facility in habitat which would allow us to breed and then release birds into the Caatinga itself. And this included a few buildings, and I'll just take you through them quickly. This part over here is the veterinary building. So you have the entrance here with washroom, accommodation, veterinary examination rooms and treatment enclosures, and a solid wall between. And on the other side, you have the nursery. So the most important thing at a breeding facility is to have a veterinary facility and a nursery facility. And so the same thing applied here, double entrance with washroom, accommodation for whoever's doing the rearing facilities. And then with an entrance on the opposite side of the building, here's the feeding area. So this is the wash room for all the bowls, kitchen where the food is produced and prepared for the birds, and then food storage, dry food storage and walk-in fridge. And this is currently where I stay. Then opening up into the breeding aviaries themselves. And these breeding aviaries include, it's in a U shape, and include 8 breeding aviaries on each side with 4 big flocking cages with 4 inside sections on each side, 2 inside sections per flocking cage. So when the 52 birds arrived from Germany and came to the facility, some of the birds were allocated for breeding and others were allocated for the flocking section, which will then join birds for release. And so this was the process and the plan of this enclosure. You have your breeding birds here and your flocking birds here. From the flocking cage, the birds would go to the release aviary, which is the next one. And from the release aviary, obviously, the doors open and allow for the birds to go out. The release aviary too was built in a U shape. And this was to allow for a large flight area, giving the birds the maximum ability to create muscle buildup, etc., and be able to learn the maneuvers and fly fast before they actually release to help against potential avian predators. The inside sections allow for us to put the birds into smaller areas first before we release them into the large areas with a communal feeding station, allowing us to then manage these birds, food supply, etc. The release aviary, the doors are on a pulley system, and what they do is they'll be opened and the birds would be allowed, it'd be a soft release, so allow the birds to come and go as they please with food inside. And the feeding stations inside will be mimicked in a few places out in the Caatinga environment that will be located in certain areas that we pick in order to give some sustenance outside during the particularly harsh dry season. But also because these birds also have to learn the wild foods. Now we are feeding them some of the wild foods in order to get them used to it, but they really need to see them out on the trees and they need to learn what they can and can't eat. And I'll get to a bit later, but we have a species that will be helping them do that. But in the meantime, we need to ensure that these birds also stay close by for monitoring in the beginning while we see the progress that they're making in the wild. We're putting up artificial nests as well as monitoring the nest cavities in order to make sure that we'll be able to monitor the birds and see what they are doing in the wild. Accommodation for volunteers to come to help with the monitoring, to help with the management of the birds. This is going to be available for students, biology students in the region, potentially for international students as well if they're interested. So once we get to the point that we're ready, and once COVID is over, this facility will then be opened up for researchers to come in and actually get involved, volunteers, people who want to learn the conservation traits from the people involved, the scientists, the professors at the universities, the local universities. Now, community involvement is crucial, and it's something that has been very important, and the Spix program has understood that concept from pretty much from the beginning. And the Curaçao community has been involved in the Spix project on a small scale since the onset of the project, the program itself. And this is very important because basically what it was doing is was fueling a desire for the birds in the population, and letting them know that, sort of branding Curaçao for the future tourism. So letting them know that tourism and that the Spix macaw can actually bring things to their environment. It can bring people from around the world, money, and thus development in the region. So this was very important. And with that, education, helping with the schools, and sponsorships with the Spix Run, football sponsorships, etc. But one of the other crucial things that a couple of organizations here in Brazil are involved in is environmental and farming management, and teaching the local farmers how to sustainably farm their livestock. Because the São Francisco River, more than 300 years ago, brought about an influx of people into the region for farming. And it's basically where the destruction of the habitat that we see here today stems from. And the increase in the number of people in the region increased the number of livestock. And that livestock, is mainly goats and sheep, has devastated the region. So teaching the farmers that they can fence off their livestock, teaching them how to produce food for their livestock cheaply, and organizing them water so that they can water their livestock and not have the livestock walking around in the creeks and destroying the most crucial Spix habitat. These are all important concepts and very important for the community to know. And to work with the community is the only way forward in a conservation project. One of the most important, and if you go away from this only knowing one thing or remembering one thing, if you work in conservation, it doesn't matter where in the world you are. If you have a project in habitat and you do not involve the community, the project will fail. Because you can't have foreigners sitting in a country running a project for decades and decades and decades and managing it. It's not financially viable. You need the community to be involved. You need the support of the community. You need the community showing that they want to be involved, that they want to protect that species themselves. And that needs to be instilled in them if they don't have it, by showing them the pros to that species being there. And if you cannot do that, or if you're not willing to do that, you may as well stop now and rather spend your money on something else, because that is the crucial part of the entire project. Everything, all the work done up to now, and all the money spent, the millions and millions spent on facilities, on research, to get the Spix to where it is. If we don't involve the community and we don't get the community's backing, it's all wasted. And the one environment that is a major concern is an arid environment, because the degradation of an arid environment is so much easier. It happens so quickly and without really just the blink of an eye, and your arid environment becomes worse and worse and degraded, so that to an almost irreversible point. And with climate change, with the temperatures increasing, it's not only this human impact, this direct human farming impact that is creating this issue, but we're now combining an international human impact of climate change. And combining these two things creates an environment that is extremely difficult to live in for humans, let alone the species that are here. And the Spix macaw, unlike the maracana, so the maracana is another species of macaw in the region. They are also endangered, but they do survive more. They're more generalists. They're able to adapt a lot better to the environment. whereas the Spix macaw seemed to not be adapting as well, and hence why the numbers were dropping over the last 300 years. So we get to the point where we have to show these farmers what they can do with their available farming methods, what research is available to help them move forward, and then helping them do it, and then showing them the positives of doing all of that. Now, I wanted to take you through a little bit of the land that we have here. So many years ago, land was bought in the region, and this is the land that we worked on, etc. We fenced off from goats. And when I arrived in 2010 into the project, I went on a trip to this land, and this is what I saw. Yes, some beautiful trees, those Cariaba trees, which is what the nesting and some of the food trees are for the Spix macaw. But you see the root system and the erosion that occurred. And this is because of the goats eating everything in their path. And the thing is with this arid zone is there's no rain for months and months and months. And then suddenly there's a short period of rainy season. And in that short period, there's generally massive thunderstorms, and the water rushes through, and these creeks flood and wash away all the topsoil because there's nothing holding that topsoil together. And basically the root systems of these trees get open to the surface, the trees fall in, and so now you have these massive trees that are nesting trees and food trees for species like the Spix macaw. You have them falling in and dying. But the problem is that all the new trees that are growing, they are being eaten by the goats. So all the small shoots are not actually getting out. So basically you're creating an extinction of the environment, the trees in the environment, because as these old ones die off, there's no new ones coming. And you can see all the feces, the goat feces in the creek system. So just fencing off the creek system does a lot for the environment. And pinyao, which is one of the foods of the Spix macaw, and the maracana and the other conures in the region, they do feast on it. And this is what it looks like once the goats have gone through it. Now, while I was out there, I got this photo of the maracana and the dam in the area, but seeing that these birds were surviving, but it wasn't a great environment for them to be in. And once we started fencing off, once we got some water supply into the region and started putting in flow-through dams, so basically what it allows is it's not a dam that closes off the river, it's a dam that just slows the water down. So it's a whole lot of rocks in a mesh case that just, as the water hits, it slows down, and it allows for pools to develop. And as these pools develop, it allows for the environment to now get the grass growth in the region, allow for something to hold the soil together. The pinyao now making bushes and available food for the birds in the wild. And there's a little picture of the pinyao itself. Cariaba trees now no longer having the goats eat them as they start sprouting, giving potential future for the species and for all the other environmental species in the region. Now, you can see over here, there's an old nest box that was put up. But this is basically a photo of the region where you start having the grasses growing into the creek system and allowing for some form of holding of the soil when the heavy rains come through. And from the work that we did, we managed to see a massive increase in the species numbers of birds in the region. Now, this was very little work. It was just fencing off the area, giving water, adding a few things here and there, a few tweaks that allowed the land to redevelop itself. We were not replanting stuff. We were not doing massive intrinsic work. We were just giving the environment the opportunity to come back. And what it did was it brought back the birds too. So we were seeing pairs pairing up. We were seeing all the different species. We were starting to see nesting again in areas that hadn't had nesting in a long time. We started seeing the large woodpeckers in the region, the ones that we hadn't seen in future trips, the ones that start the holes that end up creating nests for the wild birds. And all of this stemmed us and brought us to a point where we now have a piece of land available within a massive protected area that was announced by ICMBio and allows us to work on something. So now that we have this massive environmental protected area and we have pockets of it that are getting properly developed again, and slowly but surely educating the farmers on how to remove their goats from the creek systems and protect those actual creek systems, which are so crucial to the Spix macaw, we are now able to move forward into the actual release project for the Spix macaws. And one of the very important factors regarding this release project is the Maracana, which is another macaw within the region, and a similar size macaw, slightly smaller than the Spix macaw, and from historical data, loosely flocks with the Spix macaw, or used to flock with the Spix macaw. Now, an extinct in the wild species is very difficult to bring back. It hasn't really been done before. The Californian condor, they took the birds out of the wild and bred them up and released them again. So they were releasing birds that already knew the wild. Other birds, some curassow species, they were crossbred in order to get their species back out again. But a totally extinct in the wild species that no longer has a wild population, even in captivity. The birds in captivity are all captive birds. They have no intrinsic knowledge of their environment, and now to try and put them out in the environment creates an issue. It creates a bit of a conundrum. And this is where the idea of using the Maracana as a sort of stepping stone for the Spix in regards to education of their environment, with regards to education of food sources, predators, etc., became quite crucial. So what we did was we collected Maracana from the wild in nests, and we hand-reared them the same way we rear the Spix macaws. We then created a population in our flocking cages now that we can use as a bridge for the Spix macaw, and they will be released with the Spix macaw. But what we also did was we took an adult wild Maracana, and this was actually a crucial step, because having the hand-reared ones, we moved the hand-reared birds out into the flocking aviaries, and they had a language that they would speak. And it wasn't until we brought in the wild Maracana that we realized that they had a different language. The Maracanas had taught themselves how to communicate with each other in the hand-reared environment, but their communication wasn't the same as the wild bird. And when we brought that wild bird in, she taught them her language. And in fact, an even more important thing was, once we put her out in the flocking cage with them, we had been monitoring the wild Maracana in the region, flying around, and they never really showed much interest in the aviaries and the captive birds in the aviaries until we had that wild Maracana in there. And she, obviously, with her language, called them in. And now, every day, we have wild Maracana flying around, coming to the aviaries, communicating with the ones in the aviary. And this is creating a bridge between our captive ones and our wild ones, and the wild ones, allowing that communication to create the next step, which is when we incorporate the Spix and the Illigers in the release aviary, and we put some feeders out there and have the wild Maracanas showing them. When we open those doors, we are hoping that the wild Maracanas will sort of take them under their wing and teach them the ropes, teach them where they can get their foods, where to roost at night, and how to safely evade different predators. The one thing we did notice is that the predation, the avian predators, the Spix macaws have an innate sort of knowledge of avian predators. When an avian predator flies over the aviaries, they all duck for cover, they make the alarm calls. So we do know that they already have that, but how that will change or how that will relate to their actions in the wild to evade that predator, we're not sure yet. And that is where this important integration of Maracana with the Spix becomes really, really crucial. Because knowing that it's a predator and knowing how to evade that predator are two totally different things. The middle image here, I just wanted to show, was some bee control. We have Africanized bees in the region, and they do occupy some nests. And so we have teams going out and managing the bees, collecting them and moving them to other facilities. Now, our work in the region, our community work in the area does bring in some volunteers, some help from firefighters, etc., and also monitoring of camera traps, etc., monitoring wild nests to see what birds are in the nests. We even had Kaleba, the Brazilian power company, come out the other day to install mock power lines inside one of the aviaries. And this is in order to test the Spix macaw on these power lines that are in the region, in order to see what damage they do, see what issues they could follow for the Spix, because we know that the Lear's macaw project has problems with electrocutions. And so we want to limit it as much as possible by first seeing what the issues are and what the Spix does to these things, and see how we can then counter it. So these are little aspects, and then veterinary care, very important to have good veterinary care in the region. And so there will be a lot of work for volunteers when they want to, when they come through and when the facility is open for that. Something that is really, really crucial is the birds themselves. And I wanted you to see this photo of a couple of Spix macaws in an aviary, in a flocking aviary, so that you could see the vastness of what we have available today. And, you know, one of the crucial things is good partnerships, good healthy partnerships. And ICMBO has been a great partnership for this project. And even though we've gone through different governments, as the government changes, so staff change, there are always a few people that stay behind in order to help bridge that gap between the new government and the next government. And without those people involved, the project would have been delayed again and again and again, even worse than it has been. So that's something that's very important, is having good partnerships. And our partnerships that we have, we are very happy with and very proud of. So this is actually the side of the breeding aviary, and has a few of our partners on it. And I just wanted to say thank you to everyone for listening to what I have said, and what we're going through. And I hope you have some questions. I didn't get to everything because it's sometimes difficult to incorporate everything into a presentation. But I hope you'll have some questions that I can answer, and we can get you all the answers you need. Thank you very much.
Dr. Purchase, thank you very much for your extraordinary presentation. And yes, we have some questions here in our chat. But before I read them, I would like to make some reflections on what you just talked. If I got it right, you mentioned that the extinction of the species began about 300 years ago, right?
Well, not so much the extinction of the species, but the destruction of the environment.
Okay. Due to exploration, right, from the
area. So what happened? So a little bit over 300 years ago, there was an influx into the São Francisco River Valley, because it was incredibly fertile soil around the São Francisco River, and so agriculture became a massive thing. But from that, because of the money coming into the area, you had an expansion of people. Now expanding even beyond the São Francisco River into the communities that we now know as the Caatinga farms around the Curaçao region, with livestock. Now the people in the towns needed more meat, so you had suddenly these people on the farms had more were producing more goats, bringing in more goats, because suddenly there was money for it. There was a demand for it. So you had this boom of livestock in the region, and that is where the habitat destruction started. And from like what I said about von Spix, when he in 1819, so more than 200 years ago, when he arrived, he said it was a very rare species. He saw a very small flock. We didn't get a number, but from people doing research, it seemed that he saw about 7 birds. Yeah. Now, that is tiny in comparison to, you understand, in 1819, the population, the human population in the rural regions you wouldn't consider them to have been that great. Yet the environment was such that the species in this region were quite rare, in particular the Spix macaw. But yet Amazons do very well. In fact, when I go onto Concordia Farm, which is just down the road from the facility, in the evenings I will sit in the riverbed and I'll wait for the sun to set. And as I'm waiting for the sun to set, 60 or 70 estivas, Amazons, will come flying over and they roost in Concordia Farm, in the trees in Concordia Farm. So, you have some species that are able to manage it. They're able to adapt to the changes. And then you have other species that are not that adaptive. And the Spix macaw is obviously one of those species. And that is why it is so crucial. It is important to have got the Spix where we did, to the point where we did in the captive population to allow for a release. But the environmental protection is extremely important because we know the species is not as adaptive as the others. But the key to this is, and what I think a lot of the people in the Spix project like about it, and I think it's something that I've explained to a few people who've asked me, why do people waste so much money on a single species that has, at the point when I started, has almost no chance of getting into the wild, has almost no chance of survival? And my answer to them was, the research that we're doing is so crucial and so innovative, and it's the kind of research that no one will do unless they have a species like this. And that research is now available. The techniques are now available for other projects in the future to allow those projects not to get into the position the Spix macaw was in. And then when we do the whole protection of the environment doesn't just save the Spix macaw, it saves every single species in the Caatinga region that share the habitat with the Spix macaw. Because if you're saving it for the Spix, you're saving it for all the others. And that is the most exciting part. I'm not saving a Spix macaw, I'm saving the Caatinga. Yeah, that's my idea. That's what goes through my mind when I'm working on this project, is I'm saving an entire community of species.
That's great. And I especially appreciated the insight and the need for community involvement. It's one of the requirements for the success of conservation projects like this one. So these efforts, they make all the difference in the world. That's really breathtaking endeavor. So we do have a few questions here, and I will read them to you. Let's see, the first one. What's the perspective for the return of the species to wildlife in the São Francisco Valley region?
What is the perspective for the return? Perspective or the perspective for the other species, for other wildlife? Okay, I think that's basically just what I said to you, is that we're not just protecting the Spix. By protecting the environment, we're protecting the other species of the Caatinga too. Unless that question is regarding when we are going to release. And the first release is planned for next year. So I hopefully answered the question in one of those two answers.
The next one. Since the species went through so many disease problems, do you see some type of immunity or resistance against those diseases in the current population after all the selection process? Okay, yeah, so just to, I'll use an example, coronavirus, which creates proventricular dilation disease in the species. It is a disease endemic to the actual Brazilian region. So it is a species, a disease that, in theory, the Spix macaw should not have had as much impact as it did, or the disease shouldn't have had as much impact as it did on the Spix, because it did come from the region that the disease comes from. But we found that it did have a problem.
We had birds dying, we had birds having a lot of different symptoms. But we managed to manage it in the population, and now we're pretty clear of the disease. It might come up again, but we think that, yes, there is a bit of a sort of a form of an immunity, or they've evolved a way to cope with the disease in such a way that it no longer manifests in proventricular dilation disease. And if you look at the disease on a whole among macaw species, some birds actually get PDD while others don't, even though they have borne a virus in their system. So it is something that seems to affect birds that are immune compromised, whether it's because of genetic bottleneck, or whether it's because of another underlying disease, and this then comes up sort of like tuberculosis in humans. You know, you can have tuberculosis in your lungs for 50 years and it never does anything. But the minute you combine, say, for example, HIV, suddenly you have massive tuberculosis, or massive tuberculosis deaths in the region, and that's what we're seeing in Africa. So I think it's quite a similar aspect was found in the Spix, and yes, we seem to have gotten over that bridge, touch wood. So hopefully, yes, they have an immunity.
Okay, thank you. Let's go to the next. What are the main challenges to Spix's macaw reintroduction in nature? Lots of them. So firstly, because it's a species that is totally extinct in the wild, one of the big challenges is getting them to integrate properly into the wild. Now, it is an arid environment. So if you're releasing a bird into an environment that has plenty of food for all the species in it, it makes it a lot easier. But when you're introducing a bird into an environment that has droughts on a regular basis and has a very long dry season with very little food availability, you need to make sure that they learn where they can get their food. And this is one of the reasons why we'll be supplement feeding for the first few years until the birds have integrated properly into the wild. So that is, I guess, one of the key aspects. Another one, I guess, is related to the bees. So we have a massive bee problem in the region. So we're dealing with that at the moment, collecting the bees from the nests and creating alternative nests for the bees. So this is also something that we had to work on. And then it's predator avoidance. We've noticed that they know what a bird of prey is. We notice it from seeing their behavior in the aviaries. And again, like I said in the presentation, knowing it's a predator and being able to avoid the predator are two different things. And that's why we're hoping the Maracana will integrate well with the Spix and teach them and help them through that. If that works, we should be on a good footing. The problem is there's not only avian predators. There's also boa constrictors, which will possums, which will take on the chicks in the nest and potentially the adults that are in the nest with the chicks. So, there are multiple issues, and we will deal with all of them as we can, as best we can. Thank you. Let's move to the next. Were the birds negatively impacted when moved from Qatar to Germany, considering the climatic variation from one place to another?
Yes, it always happens when you have a big move, and we noticed it because we were producing 20 odd chicks a year, and then the season that we did the move, we only had 11 chicks that season. So yes, you can see the impact, but that impact is related to moving them during the breeding season. And then regarding the weather conditions, the facilities in Germany are climate controlled. So yes, the outside aviaries have the cold weather in winter, but they do have the option of being inside where the temperature is controlled and managed for the birds. So temperature-wise, I don't think it was that much of a change for them or that much of an impact on them, even though it was a massive change.
Let's move to the next. We have lots of questions if we have time.
We have plenty of time. Fortunately, I'm not used to talking for long, so we have a lot of time.
Yeah. So here's the next. Is there any plan for planting species in the release area that are favorable to the environmental enrichment of the macaws?
Okay, so when the bird's in the wild, enrichment is really the wild environment itself. So not particularly for enrichment purposes, but there will definitely be planting of Caatinga species, and in particular species that have become extinct and have become very rare in the region because of the livestock farming. So yes, we will be planting food trees, nesting trees, etc.
Yeah, thank you. Next, in general, is the perspective regarding the future of the macaws good, considering the intensification of works like this?
I like to be positive. I like to think that the future of birds are, macaws in particular, are on the up. Obviously, the environment is a problem, and the climate issues that we're having and the increase in human populations is obviously creating issues, not just for macaws, but for wild species around the world. But there are quite a lot of projects at the moment that work with macaws. The Green-winged macaw project, the Buffin's macaw project, scarlet macaws, so and then Lear's macaws as well. So it is something that's increasing. There seems to be a lot more conservation work going on now in situ than there have been in the years before. So I think it's a positive thing.
So we have just a last one, and it's quite an interesting question. If I'm a biology student and I want to help the project, what can I do?
Okay, biology student from where?
Oh, he's a colleague of ours from here.
Is that why you were smiling?
Yeah, it's from here.
Okay. Yes. So there are a lot of possibilities. We are waiting for the construction to end of our volunteer facility, accommodation facility. Once that is done, we will open it up for students to come in. So it'll be students from the university, the local universities, and we'll also allow international students, anyone that's really interested in working on the project and has a keen interest in learning the different protocols and things that we go through in conservation and for a species release, but not just the release, but also the captive environment, because we have a breeding facility as well, learning the whole captive environment and how to manage a captive environment for the wild. There is a lot to be done. There's a lot of work involved. We got a lot of work from ICMBio and people getting involved. So there is so much to do, and there will be hopefully a lot of people coming to help do it. So what can I do? You can keep us in mind. Just ACTP is our organization, and on our website, as soon as the volunteer facility is available, the ACTP website will have a link to applications for volunteering.
That's great. I'm sure a lot of our colleagues will be paying attention to that. Thank you.
Yeah, you're close by, so it'll be easy for your guys to come in.
Yeah. We've reached the end of our questions, but we have another comment from a very important viewer, and it's Claudia, who was asking to tell us how are the babies.
The babies are good. They are now eating on their own. So we have the first baby's been eating on his own for a while. The two others that we have there at the facility they've just started eating on their own. So in the next few weeks, they'll be going out into the first aviary, and from the first aviary, they'll then go out into the flocking aviary, and then hopefully in the near future, they will be in the release aviary.
So thank you. I think that's all of the questions we had. Dr. Purchase, thank you very much for being with us today. It's been an honor, and it's very important to have you here in our 9th Biology Week. I'm sure a lot of our colleagues and all our viewers have greatly appreciated your presentation. So thank you, and if you want to say anything we can finish the presentation, the floor is yours.
Thank you very much. Thank you for having me. It was awesome to be involved, and I hope my message got out and that people are interested in coming and helping with the project. It would be really, really awesome to see people around that have similar interests in conservation and working on these kinds of projects. Thank you very much.
Thank you. Thank you. So we'll finish this presentation, and I thank Dr. Purchase again, all our viewers, and please follow up our next activities, and that's it. See you.
Cheers.